will make this slightly smaller. It's a bit big. This is probably good.
Yeah, is everything fine? Has everyone this running? And yeah, feel free to ask questions. I think I might not be clear on everything, but I will do my best. So I hope you were able to run this cell and you got this message so that Hail is running and everything is fine. So the next steps are pretty simple. So these two we specify the path to the data files. So these files are available already on Databricks, and it is a small subset of human 1000 genome project. So basically, it just subsampled to be able to run it quickly in Databricks. But if you're interested, you can of course download the full data set. So let's run this cell. Yeah, the files, the paths now are saved, these values. And the first step will be to import the VCF file. This is the biggest file of the two we have, so it may take some time. So here I wrote that usually it imports already compressed file BGZ. I have tried to do it on my own with my own data. I don't know, maybe there are different compressor versions, but sometimes it doesn't work. So if it doesn't work, you can also load uncompressed data. It will still load it, it will detect it automatically. So if you want to use it in the future, you can load uncompressed data too if something wrong with compression. So here it just shows the first few lines of this file. It's quite difficult, I think, for human reading, but as you can see, there is a lot of similar names here. So basically, these are the column names. So these are different samples. This step is not maybe necessary for this data set, but what? Let's see, now we have the first error here. I don't know why it complains. We have VDS here. Yeah, that's what happened. But I checked it yesterday, it worked on my computer.
It's working here.
It's working for you?
Yeah.
I don't know why it doesn't work. Yeah, for some reason it doesn't have split function.
Oh, yeah.
Let's see, I think it's possible that there are some other libraries which automatically attach to this cluster. Yeah, you have.
Yeah, maybe detach the others and
there may be some conflict. How can I detach them all? I can do that from here. Yeah, because we have two Hail here.
Oh. Yeah.
Can I detach them from here?
Yeah, just go to that Hail, that one, the shared Hail jar.
I think I want to remove this one. Yeah, I think if I do this.
Yeah.
Yeah, so but if you attach only one, I think that's why it works correctly for you. These are also not necessary, they were just configured before by us.
So you need to restart the cluster now.
Yeah. Yeah, but this is good. So now we know that we may have some problems. Yeah, but basically what that step does, if you remember us was looking here that you have 2 values per site, and this is the most common case. In most positions you will have like 2 values at maximum, or 1 if it's fixed, for example, 1 of the alleles. But sometimes you may have 3 alleles. It happens, usually it's very few sites, but most of the models, they will assume allelic, like biallelic sites. So to fix that issue, if you have some sites with more than 2 alleles, you run this split multi. So I need to split that site into 2 sites to keep only 2 alleles. So some sites will be duplicated to keep only 2 alleles. Or if you want, you can also remove those.
I think you have to restart the cluster, right? Because
I have previously started.
you did. I thought you detached and then restart.
Did I click
Right?
attach?
So let's try. So let's see. Still works.
And you don't need to run. I think it's saved, no? Even if you restart, it's written already.
Yes, I don't think you restarted.
No?
I think you did detach.
Yeah, if you restart, you can restart again.
But then your cluster, I mean your jar needs to be reattached. Right?
No, I think it reattached it automatically. Right? Yeah, I can check. It's fine. It's yeah, sorry, I just need to run everything again. Yeah, it works. So what we can do here, we can just run everything above this cell. So it will execute everything until this cell, and then we can start from this cell again. Yeah, probably you have a jar file loaded from the older version, which is in Databricks, and that doesn't work with this function because I think it was written later. Yeah, now everything works. Yeah, but as you can see, there is a message that all the multi-allelic sites, they have been split already. But if you start with new dataset, you may need to run it. So just to show. So here we import the annotation table. So we have a VCF file with the columns like different sample names, but we also may have additional information for every sample. For example, in this case, we will have information on their location, like different countries and different regions, or you may have a phenotypic data. So if we scroll down here, so this is annotation, and if you click, you can display it. It's already displayed in my case. Yeah, one more point here, but I think you probably know it. This display function, it's Databricks specific. So if you run it not on Databricks, you will need to use other things like print or something, I think, to print this table. But here you can see this is a sample name, and you have seen these names when we loaded the VCF file. And then we have different columns for each sample, like population. Then we have superpopulation. I think this may be like Great Britain or I don't know, some country, but it's in Europe. And if we scroll this table, I think we should be able to see some other regions at some point. Yeah, so here we have Eastern Asia probably. Then we have a gender here and some phenotypic data. We're not gonna use this phenotypic data, but you can use it for some analysis.
This purple hair.
Yeah, what is that?
That's color hair. That's one of the phenotypic data. I don't know. Maybe they invented it.
I have no idea.
You can I think there is a blog post on these 1,000 genomes. You can read more. I read it last year, so I don't remember what was exactly there. So now we can kind of see how this annotation table is applied to our data set. So this is the sample schema. So this is annotation of columns. Oh, it says it's empty. Did I skip some parts? Yeah, I skipped this one, sorry. Yeah, first you need to annotate the samples. And after they are annotated, so we annotate these samples with the table we have just seen. So basically we add this annotation on top of the columns. And then you can see all this population, and you know that it's a string. Then we have superpopulation, and gender, you can see it's Boolean, so it's male or female, the same for the hair and coffee's consumption. You can also print a more extensive format of this annotation schema, like it's a JSON format. So you can see a bit more information. So this is the population, this is string, I can some other parameters. This is where it gets difficult, like for person like me, because it's a bit more abstract presentation of the data. But this is how it's done, and you can do the same for variant, but yeah. So invariant annotation is annotation by the rows. So samples is by the column and variant is by the rows. So for example, here, just like if we have any filter applied, we can see it. Like, for example, this is allele frequency. So if we have one position, we can have the frequency of, because we have two alleles, so one of them may be more frequent than the other, so we can get this information for every row. And a lot of other information, you will see it later when we do filtering by variants, we will use this information. Yeah, and here I just show some sample, some simple statistics you want to know about your data. So the one is how many samples we have. I mentioned before that there are 1,000 genomes project, but this is subsample, and in this subsample we have a little more than 700 samples. And what we can also do using the annotation schema, we can check how many samples we have per population. So if you run this, by default, I think it may print you a table, but if you click here on plot options, and you use population as a key and values as a count, and cannot scroll it for some reason. But here, a little bit down, you can select histogram, and you will get this plot. But I think if you use my notebook, it will show you this by default. So you can see that most of the populations are represented more or less evenly, on average like 20 individuals per population. So this is good. And we can also get some summary statistics about our genotype data, about this VCF file. So if you want to get everything, you just run summarize, and it will print you everything that summarize function does. So in this order, it gives you number of samples, number of variants. So we have checked it before. We have 710 samples, and each of these samples is represented by almost 11,000 variants. So it's almost 11,000 rows. And you can see that most of the genotypes are called successfully. It's only 2% non-called. We have 23 chromosomes because it's a human data. Yeah, this number of SNPs and number of variants is the same. If we had some sites which are not variable, so if all letters are the same, we would see some difference here between these two. But you can simplify your life here if you save this summarized results as value like some VDS. And here I have a small assignment for you. If you look at this object, so you need to remove these two slashes, and you put your cursor here and press tab. You can extract specific information you want. So for example, I don't know, let's use call rate, and you run it, and it will give you the call rate only. So basically, here it's printed everything, but it may be difficult to see exactly what you want because you need to follow this order. but you can access specific statistics just using this kind of additional step. Oops. So the next step, we will do some filtering, because often when you just get your VCF after genotyping, it still contains some errors or low confidence genotypes, and some samples may have a lot of missing data. So if you want to do some estimates, it makes sense to remove samples with bad quality, or you can also remove some variants. For example, if you have one variant where only like 30% have genotypes, the rest is missing data, so it can bias your estimates, because if you calculate, for example, allele frequency, it will be not very accurate if it's very small sample size relative to the others. So there are many annotations, scores, and statistics. I'm not going to explain all of them, but one simple one and widely used is genotype quality, and it is estimated with this Fred quality score. You can read more details how exactly it is estimated here in this Wikipedia page. But to have an idea, so if we have a score of 10, it means that it's 90% accuracy. If we have 20, it's 99% accuracy. So nowadays, the sequencing quality is very good, so usually you keep everything above 30, but this dataset is not the newest. So as you will see here, the scores are a little bit over here, but we can assume that 20 is a good score because you have only 1% error. And it's up to you what kind of study you do. If you do just general population genetic study, you can allow some mistakes because it will not affect you too much, but you will keep a lot of data. But if you do genome-wide association study where you're interested in specific SNP and you want to see if it's associated with particular disease or particular trait, then maybe you want to do more stringent filtering because you need the highest possible quality of the data. So this is genotype quality. It's estimated based on each cell. So what we do here, we take this VDS file and create this genotype table and transform it to data frame, and then we display it. So here where this kind of abstraction comes, so this is a variant, as you can see. So this is the first chromosome, like chromosome or contig, it's kind of the same terminology here, and this is its position. And as you can see, basically this cell and this cell is identical because it's the same position, but it's just different genotypes, because in every row we have many genotypes, so we have many columns. So here they kind of stacked one after another. And if we go and look at these column names, you can see this is a variant quality, but that is not what we need. We need genotype quality. We go to the letter G here. And here you can see genotype quality, it's 12 here, like GQ, this is the genotype quality, and DP is depth. So depth, it means how many reads you have for that position. So in the first cell it's 4, so it's not very good. As you see, we have this reference position and we have only 4 reads. So if we had 30, well, it would be higher confidence, but it's only 4. So not the highest, but if you look at others, you can see here we have 8, like 5, and other. So what we're gonna do, because it's a genotype, so if you type genotype.dp, you will refer to the depth of a particular genotype in your dataset. In this example, I will use only depth, so it's number of reads per that position, and genotype quality. So how confident we can be, how accurate the genotype. So here we will use some SQL. So we just create a temporary kind of file. I don't know, this is Evgen, he was helping me with this. And then we select these 2 variables. We select depth and select genotype quality and save them into table as genotype quality and depth, like you will see here, from genotypes, and genotypes were created here. Yeah, and we also applied some filters here, so we skipped all those which are missing data, because they will take extra space. And with Databricks, it was also difficult because the table is very big, so Databricks could not handle it. So we additionally randomly sampled only 1% of the data and limited our output only to 1,000 lines. But this is enough to get a general picture of the distribution of different scores. And when you know the distribution of different scores, you can filter. You can see where majority of your data is, and then you keep that part. But if you have the tail of this distribution, you can remove it.
You can run the previous level.
Probably this one.
Yeah.
Yeah, by default, Databricks outputs you the table with the function display, but you can also modify it. For example, if you click here, you can see the plot options, and you can make a plot. I have done it here in the next cell, but it memorizes the previous configurations. That's why it shows this plot, but it's also possible to change it to table. So it's the same table, but just present it as a plot. And if it looks like this, you can always stretch it. Yeah, so this is how it looks. So this is a distribution of genotype quality, and if you remember that Wikipedia page, I said that everything above 20 is pretty good. It will be only like it will be 99% accuracy, but this data is not the newest, and at that time the sequencing technology was not very good, so we still have some not very good genotype quality, but we can define a threshold, for example, like 10 or 15, so we can remove everything of not very good quality from here and keep everything of good quality. We have some very good genotypes, but as you see, majority of the data is here. But if it was modern data, that would be like this probably. Most of the data will be about 30. So here I have a small task for you. You need to do the same, but for the depth. So it is simple because we have created the table before, right? So we extracted depth and genotype quality, but now you need to make a plot for the depth. Oh. Yeah, try to do it like for 1 minute. So it's simple, you just need to execute the same command. So here we have depth and genotype quality, and you need to build the histogram for this column. So you click here, this is how it looks by default, but it's not what we want. And if you go here, you can remove this ID. Why can I see this part? Yeah, I will decrease it just to see it here. And if you select histogram plot here, you will get a distribution, and you can define the bin size, for example, 30. I don't know, even 100, just to see how it looks. Yeah, 100 probably too much.
You also want the values to be depth.
Yeah, so you can see that on average, most of the genotypes are covered with 20. Yeah, so you can remove these two columns, for example, which are not covered too much, keep most of the data. So that's what I wrote here.
You want the x-axis to be depth.
Oh, didn't I change it? Okay, yeah, sorry. Okay, yeah, that's different, you're right. Yeah, so the coverage is not super good. It's less than I expected. Because now, like even in this Adam talk, they say when you sequence use human genome for $1,000, you get coverage 30 on average. But as I said, this 1,000 genome project, it's considerably old. And as you can see, most of the reads are covered with 5. Most of the positions are covered with 5 reads.
This is 2015. When did they do this? 1,000 genomes?
No, I think it's 14 or
2014.
but I don't remember exactly. But sequencing technology improves a lot. When I started my PhD, I had one data set, and like 2-3 years after, I sequenced another data set, and there was dramatic improvement. Like I even didn't have to filter a lot after that. And I think they keep improving, and it's getting better and better. So here I suggest to filter for reads with less than for genotypes which have less than 2 reads and quality less than 10, they will be removed. But keep in mind when you do the filtering, you specify what you want to keep in this case, because it depends on the program you use. Sometimes you specify what you want to remove, but in this case we specify what we want to keep. So if you run this one, so this is a built-in function, filter genotypes, and then we specify these 2 parameters you want to apply. You can also use or, for example, here, not necessarily and. I don't know, I think if you type it this way, yeah, so it will be either of 2 should be true, but we are interested in both of these parameters, and you can use as many parameters as you want. There are some other annotation statistics. So now we have removed some cells. So this filter was applied for a particular cell. So you take particular position on the genome and particular sample, and you check the coverage for that individual in that position and the genotype quality. When we remove some cells, we get missing data in our table. So now the call rate will go down because we introduced some additional missing data. So we remove some genotypes. Here we check the call rate. So you already know the summarize function. And we just want to see what's the call rate of this new data set. And you see it was 98% before, now it's 83%. So we have removed some data. So the next filter we can do, we can remove some columns. For example, if some samples were sequenced not very good, and many genotypes were removed with our previous filter, maybe it makes sense to remove that sample if it has a lot of missing data. But because the sample annotation was created on the previous data, we need to redo the annotation again because we introduced missing data. So let's re-evaluate these different statistics for our samples. So this is sample QC, it's a quality evaluation of different samples on this new data set. So basically we kind of update our sample annotation for every column. So when you have done that, we can check this annotation data. So here we use sample key table. Previously it was genotype key table. And again, you just pattern it because there's different arrays, and you create a data frame from it. And we display it with Databricks tools.
So are the nucleotides of these positions there in the data frame?
No, this is annotation for samples. So you have summary, like for example, the number of code positions per sample. For example, here you can see this is the number of positions which are not code, so missing data. This is number of code positions. This is a number of homozygotes which are the same as reference. This is heterozygotes. We can also have number of variable position in different samples. Like you can see, there are many statistics. Like we can have mean genotype quality, for example, or mean coverage, because we filtered for the depth before. So now, as you can see here, most of this is about 5, and I think we filtered for 2. So everything below 2 was removed. So this mean values should be improved. And again, of course, it depends on the data and your question. You can filter it differently. For example, we can see the mean call rate for different samples.
I get some errors when I try to do the VBS things. Yeah, you have to do the same.
The same?
You have to reload according to the instructions, right?
Yeah. Have you restarted your cluster after the first two cells, 2 bytes?
No, not sure.
But what's the error message? Let me see.
I could run the things about that. And restart them.
It says drop and board. I don't know. Can you run it again? This error, yeah. So you need to run the first 2. Or you just need to restart. I think you ran it before, but if you do it here and restart.
Yeah, I just did. So. But
now you need to run everything above. But you can run from here. If you click here, you can see run everything above.
Yeah, that's true.
Yeah, if you get error message saying something like Java, something was not found, that's because you have not restarted the cluster. The first 2 cells, they were to correct this problem. So here again, you can see that most of the samples have a pretty good call rate. So majority of the data has more than 80% of their positions with genotypes and some missing data. But as you can see, some samples are very bad. For example, here we have so this one column, and only 30% of this column is filled with the data. The rest is missing data. So it makes sense to remove those. And I don't remember what I choose here, but let's say 65 or 70. Like everything below this, you can remove these samples. So here we remove columns. Previously we filtered different cells. Now we remove columns. Yeah, so I decided to keep everything above 60. So I will keep only the samples which have 60% or more genotypes. Did it work? Yeah. So we had 710 samples before. Let's check how many samples are left after our filtering. Yeah, now we have only 667 samples, so we removed some proportion of our data. Also, it's good, especially if you use this program first time, it's good practice to check the distribution after the filtering, because if you type the wrong sign, you can actually remove what you wanted to keep. So if you run this, if you collect this statistics from your filter data set and make a plot, you can see that we start at 60 here. Yeah, here 60. So everything below 60 was removed, and this is what we wanted. So we kept the majority of our data. And if you go to these plot options, you can check. You can change you can check different statistics here if you want. Like you can go for mean depth, for example. And it will change. You see, we have everything above 5 because we filtered for 5 reads per genotype. Yeah, that's what I suggested you to do here, but I think we don't have much time left, so I will skip that. But if you go to the settings of the plot, you can change different variables. So now we have removed specific cells, we have filtered for bad samples, and the last step we can do, we can remove the bad variants. So here I refer to JTK best practices. If you want, you can read this, especially if you're interested in doing this kind of filtering for your project. There are some commonly known thresholds for different statistics, what you keep, especially if it's human data. It's very easy because it's widely used. So you can use just those. I will use only some of them. Yeah, so I didn't say at this step we re-estimate the quality of our variance. Because we changed the table, so the annotation of these variants needs to be updated because we removed some samples and we also removed some genotypes. So you evaluate different statistics for every row and you extract that information to a separate table. Here principle is the same, but we use variant key table here. So we have used genotype key table, sample key table, and now we use variant key table. And you just create a data frame from the table. So as you can see here, again, you can just look at the column names and see different available statistics for variants. So variant is VA, and you can see, so here we have allele frequency, base quality, rank sum. So if you're familiar with this, you probably know these statistics, but the simplest for us to know, for example, depth. This is the depth. No. This depth is different from the previous depth because previously it was depth for each position, but this is the depth for the whole variant. So this is across all positions. Yeah, you can see we have many different things here. For example, like mapping quality, this is very useful. If you want to, some positions in the genome, they are very difficult to align. So if there is some complexity or some variation, when you map the reads, they just tend to purely map to that region. So that variant will have low confidence. If it has low mapping quality, you want to remove that variant because you will have false positives in that region. So if you display this table, as I have done here, so this is quality by depth. This is another parameter because these two are connected. The more reads you have mapped to a specific position, the better quality will be. So you need to use these two parameters together. But you can explore some other statistics here. If you go, I don't know, let's just yeah. Where was I? Here? Yeah, sorry, jump somewhere. Yeah, but you can look at different variables in that graph. Is everything fine so far? Feel free to ask questions. I think I may go too fast, but I just want to finish it. Yeah, so I was here. If you go to plot options, you can change this quality by depth with some other. For example, mapping quality. And you see this is a distribution for different variants. So we have already filtered it, I think.
Is this one recombinational mapping or is it mapping to the reference?
This is mapping to the reference. This is evaluation of mapping quality for particular variant. So it looks across all genotypes, what was the mapping quality, and estimates how confident you can be in this variant. For example, if you have like very symptomatic region, which have many repeats, that will be very difficult to map, so the mapping quality will deteriorate in that region. Yeah, that's what I suggested here. You can check mapping quality. That's what I have just shown. And I have hidden the results here, the answers here, so you can click and look at them. Yeah, we actually need to run this. So here, it's a similar principle as before. We filter different samples, but now instead of filter samples, we filter variants. And we use some expression language here. So we filter for mapping quality and quality by depth. And these thresholds just defined based on the distribution. You look where majority of your data is and try to remove kind of a tail of the distribution, because it's a good trade-off. Unless your data is very bad, sometimes you may see that all your genotypes are, all your variants are bad, so then you have another problem. And if we run again, so this, oh, I think I have not run this one, right? Yeah, so if I evaluate this one, so we have removed some samples, but as you can see, we have not removed a lot. There was 10,961 variants before, and now it's 10,660 variants. So this is fine because we have removed some bad samples. Because we removed bad samples, we actually have not affected our variants too much. We just remove few of them. Yeah, again, here you can plot the scores after the filtering. So as you can see here, everything below 5 was removed. I will just skip this part. We just evaluate our filtering criteria. So after you have done all your filters and you have created the database you actually have some confidence in, you can save this database into a file for future references or future work. Like usually when we loaded it was VCF file, so it was just a genotype data, but here it's VDS, so it's variant dataset. So this file, it also contains all the annotation, which we applied to our VCF file. So it's not just a text file, it actually saves if, kind of several files per that dataset, if you check. And then in the future, you can just load this dataset and do any analysis you want. So we just read this dataset again. So now we have loaded it, and as an example, we can explore if there is any structure in our data. One of the ways to do it will be to perform a principal component analysis. So this is a built-in function in Hail. If you go to Hail documentation, you can read exactly what kind of algorithms they use for this particular PCA. But basically what it tries to do, like in this particular case, we keep only first two principal components. So every variant will be kind of a principal component, and you perform the rotation to reduce variation as much as possible. And we build and we visualize the first principal components to see the structure of our data. Yeah, so when we have done principal component analysis, it has added the annotation to our samples. And this annotation just two principal components. So let's have a look at this at the scores of these two principal components. Again, we extracted that annotation as we have done before, this sample key table, and we display it as a table using Databricks display function. I hope you are familiar with this table already, you have seen it before, but if you go to the end of this table, so here every row is a sample. So you see this is a sample name, and you remember some of them from Europe, other samples from other regions. And then we have a principal component scores assigned to each of these samples. So this is PC1 and PC2. Next, we need to extract these two principal components and make a plot. This is when the problems sometimes arise. If you want to use it in SQL, SQL will not recognize names with dots because in this table you see the names are separated with dots. This is sample, quality, and then we have like 3 words here, they just separated with dot. If you try to extract it with SQL, it will not work. It will complain it doesn't recognize it. So what we're gonna do here, we just take these columns and replace each dot with underscore. So this will be a new table which has renamed columns, and as you can see in this table, here we have underscore for every name instead of a dot. So now we can use this table for SQL extraction. So here the approach is the same as we used before. We just extract superpopulation, so it's Europe, I know, Africa and other regions as population, and PC1 and PC2 as PC1 and PC2. Okay, I think we are on good time. After that extraction, you obtain this table, but you can extract whatever information you want. For this example, I simplify just to 3 columns, and we can make a plot. One of the ways to use the Databricks built-in function display. And if you go to plot option, you can see how I done it. So I plot PC1 and PC2 with population as a key, and you see this my keys. And here is the structure for our data. As you can see, we can find quite clear separation between different regions. So there are at least like 4 big clusters, and I think if you look closer, you can find even smaller clusters here. So what it tells us that our data is not homogeneous. So most of these guys will mate with each other, and like they do mate and this with each other. So there is clear population structure, and if you want to estimate some kind of coalescent, if you want to get coalescent estimation on this data, you will get some problems because these two, like these guys, will coalesce more like faster with each other than these other populations. Of course, there is some migration and other patterns, but this is good to know. If you have a strong population structure in your data, it helps in further analysis and interpretation of your results. So you can.
The purple one is what African samples, is it? AFR, probably African.
I'm not very good distinguishing colors.
You
but
are,
yeah.
I think it's European.
This is African, right?
Yeah, it's African, yeah.
Yeah. So this is Europe. This is
American?
East Asia, probably.
Yeah. A1 must be American.
Yeah, American is between, you see here.
East Asian. Oh, SA is South Asian.
Yeah, maybe South Asian.
And
yeah. If you go to that human genome notebook, you can download it. It's in archive now, but you can read all the information there. Do we have anyone working with human data here? No? Yeah, if we had someone, I think they could help us. You can.
I mean, that's sort of kind of consistent with the out of Africa hypothesis, right? So humans supposed to be came out of Africa.
Yeah, because you see the first
Eurasia.
The first PC is the strongest one, and it's separating Africa from the other.
Yeah. So.
I haven't checked. I think you can also extract the percentage, like the load of different PCs, but I don't have this information here. But if you, I think there must be options when you run PC to extract the information too, so you can actually see how much variance explained by PC1 and PC2. That would also help. But PC1 is much stronger than PC2, and Africa is the most distinct from all the other populations. Yeah, so this is your home assignment to do the same for PC3 and PC4. You can see how much structure maintained in kind of smaller principal components. If you want, like if you run it not on Databricks, but on your own server, or if you run it even locally, you may be interested to make a plot, but display will not work because display is proprietary function of Databricks. But you can run R code. Here you use the same data, you just extract it with SQL within R. So you can just use SQL function in R and create a data frame. So okay, let's run it. So it's the same what we have done before, but this time we save it in R. So this is also very good because you can jump between different languages, and you can use a plot function of your liking. You can use ggplot or just even simple plot like standard R library to do plot. And here I changed it a little bit. Previously it was superpopulation here, so by different regions, and here I went to a population, so it's a bit more kind of partitioned. But you can see again, so even within different countries, we have some strong clustering with some more distribution. Yeah, but I think you need annotation to decode all these names. Yeah, this one, I think it's some African. Yeah, so to summarize this notebook, basically it shows how to do some standard filtering of your data to get high confidence dataset for future analysis. You don't need to filter exactly the way I filtered. It depends on your priority. Usually the first step is to filter genotypes. So you remove specifics, like you replace specific cells with missing data if the confidence in the genotype is low. And then based on what's your priority, either to keep as many variants as possible or to keep as many samples as possible, you can first filter samples and then variants, or you can do the reverse. You can remove some variants, and the later step will be to remove the samples. Yeah, and when you have this data set, you can save it and use it in any other analysis in the future. You need to go to Hail documentation to see all the possibilities of this software. They have some useful functions, but not a lot. Like if you really want, you can extract some additional information into save them into files. For example, we were interested to export genotypes with RAS to get this kind of this matrix for you and do some analysis. It was a little bit tricky because the way I think the data set is written to a disk is different from what we think. It's not written as a table, because if you do the extraction, you can see it extracts first position. So basically it takes the first row and it prints it. Then it takes the second row and it prints it after that part. So this is the same position, but different individuals. So this is chromosome 1, position 4,165. And then we have genotype, so we have some missing data for some individuals. And if we scroll, so we need to scroll like 660 or something. Yeah, so
it's for the same position? Or
yeah, it's for the same position for different individuals. And if you scroll long enough, you will reach another position after this. Yeah, here's the next position. So it starts this way, and in Adam, like they create these Parquet files, it's written the same way. I don't know, they explain it, this is how the data is written to a disk. Yeah. For some reason, if it's written this way, then it's faster access or something. I don't know, so then computer way, because that was difficult for me when I started to read all this documentation. It's not the way how we think about tables and everything. It's a bit more abstract, and it's kind of, you have this data set and then you have a schema which defines how to access different parts of the data.
I mean, because this is too small, we could potentially do this deadly thing called group by key. So we can group by the position, and then it will create, remember group by key was one of our basic operations on RDBs. Then it'll actually put all those the nucleotide variants in a big array for that position.
Yeah.
So we could kind of do something like that, or write our own group by key or reduce by key function where we write a little reduce operation that will essentially build this vector for us. There must be some standard way these guys should go to the binary instance matrix world.
Yeah, there might be some other function. As I said, I just started to learn this project, and I have never done any like real life project in Databricks and in Hail, so it's still kind of new information for me. But here the genotypes are coded a little bit differently. If you have 0, it means it's homozygote for the reference. If it's 1, it's heterozygote with 1 alternative allele. 2, it means that it's homozygote for alternative allele. So you see, this is the coding. So if you want to extract this information for the future, but you can also get some useful statistics also already using Hail, like for example allele frequency or non-reference allele count. Like here, I have extracted allele frequency, and then how many alleles are different from the reference, for example. And you can also get correlate, for example. That's we have looked at before, but this is correlate for different positions, not for different samples. Again, when you have a table, you can make different plots. So I have created a table of allele frequency. Here, as you can see, it has some kind of elevation in the middle, but this is probably because of the population structure. I think usually you would expect some more kind of decay here. But because our sample is not homogeneous, there is some population structure, so we have some elevation here. Yeah, so this was the first notebook. Yeah, and this is the frequency of non-reference. In Hail, they also have a tutorial for genome-wide association study. You can use that if you go to the Hail documentation, but there was another package published also recently, and they claim it's better than Hail. So it uses random forest to perform the association. For those of you who don't know what genome-wide association is, I don't know, I found this video. It seems to be clear for me. So yeah, it's quite short, just 2 minutes, so let's watch it and get some idea about genome association. And then I will explain more details about this.
We conduct something called a genetic association. Now, these studies take people with a trait, for example, a skin condition. Really, they take a whole population of people and they compare their genetics to an individual or really a group of individuals without this particular trait, in this case, without the skin condition. Now, what we're going to do is look at a genetic factor or more, and we're going to see which variant each population has. Now, if a certain genetic variant is observed more frequently than you would expect by chance.
Here it's abstract, you see it's BLT, but you can, in our case, it's ATGC, like 4 letters, and they can be associated with different traits.
In the actual cases and control, there's good evidence that it might be involved in the trait. Suppose that we have 1,000 people that have a particular kind of skin, and we compare them to 1,000 people that don't have this particular kind of skin. Now let's say we analyze these two populations for one particular genetic factor. And let's say we determine that 50% of the population of the skin has a cytosine for this genetic factor in a given position. And then in the control population, only 20% has a cytosine. And with that same position, the only other option we can have is an adenine. In our affected population, the other 50%, the remaining 50% have an adenine. And in our control population, the remaining, this is 80%. Now we have a lot of people in our population, thousands. If this letter was insignificant to this particular trait, we would expect that for so many people, that these numbers would be more similar, 50-50, 50-50, or 28-28, both. In other words, these differences are greater than what we would expect just by chance. So based on the scenario here, I want you to tell me which of the following is most reasonable conclusion.
Yeah, so as you can see, if you have some phenotypic data,
We want to import that number, of course.
yeah, so let's import that. Close database. So it's on the same page, the previous notebook. So again, it's here. You just copy, you open this link, and import it to your notebook. I don't know. It's still not much time left, but I don't want to go too fast to explain it. But basically, we have these different positions in the genome, and let's say we have a phenotype. And let's assume that, like, if you have two groups, usually like you have one group and a control. For example, you have one trait and here you have a zero trait. And you look at like chromosome 1, position 1. And in this simple example, you have 5 individuals in this position. So we have, let's say, A. And then you have another group, this phenotype 0. I don't know, it can be hair color, for example, black, like blonde and dark hair. And here you have completely different genotype for 5 individuals. So, but this is kind of an ideal case. So we have a clear separation between two groups, and when you run across many different positions, you can find some of these associations. So you have many positions. But it's not always like that. Sometimes you will have some different, for example, in this case it's like this, but here, let's say you have different variation. You have 50 and then you have 8. But here you have, let's say, more 8. And then you have this stitch. And this kind of algorithm here, and I'm not very good maybe explaining this random forest, but it randomly samples different positions and makes this decision tree, like splitting this dataset based on phenotype, because you know the phenotype for each individual here and each individual here, so you know this is 0, this is 1. So if you have clear split here, but you may not have clear split here, but we can still get that T is more associated with 1 and A is more associated with 0 in this position. But this is a small example. If we have like 1,000 of the genome, we have a lot of genotypes. So we have some mutation maybe in some individuals, but in the genomics, the sites may not be completely independent. So if we have some mutation here, the same sequence here, they will act differently if there was no mutation. So this is kind of biology, not a math part. And based on this association, you can estimate how much association you have per position. So this is so-called Manhattan plot. So
can you please take down the...
Yeah. So here the x-axis is the genomic position. You can see these are different chromosomes colored in different color, and this is p-values. So you have some genomic kind of background values, as you can see here. On average, you have these p-values, but then you have few positions which have very high p-value. So this particular site variation in this particular site has very strong association with phenotypic variation. So if you have two groups with different phenotypes, in that particular site, you will be, in most cases, very close to this kind of association. I have never done genome-wide association study myself. This is actually was the first time I tried to do it. So here they give the information about the data. Again, it's 1000 genome project. And for the phenotypic data, they used so-called hipster index. Actually, you can go to the original blog post. I leave a link here. Did it open it in new tab? No? Yeah, but we can quickly have a look. So you can read about the background, why they decided to do it. But basically, this approach with random forest, it was more efficient. And here is some graphical information about how it works. So these again different positions, and these are different samples. And then you randomly sample different variants and make a decision tree based on the phenotypes. As you can see here, so you have one phenotype, another phenotype, one phenotype, another phenotype, and you build many of these trees, and based on that you can kind of define, for example, healthy or sick persons, or the same you can use like not necessarily disease, but some other traits. And it seems to be very efficient, so it works very fast, as you can see, in comparison to the previous libraries, and it also much more powerful. And here it's also more accurate and faster again. But this is more math part. It's not easy for me to explain everything. But what's funny here, they used these few traits, which they define as a hipster index. So they use the phenotype as monobrow, then the beard. I don't know how they define it, how well it grows probably. Then for the short, they used actually the variation in our kind of eye retina about the perception of color, and they also estimated the coffee consumption. So if you drink a lot of coffee, wear this squared bright t-shirt and have monobrow, this beard, so you're probably hipster. And based on the variation of these traits, they estimated so-called hipster index. But these are kind of a real genomic trait, I believe that was my understanding. I don't know, should we create a cluster and run everything?
Yeah, take your time, 20 more minutes, right?
Yeah, so here you will have exact position, because they already selected the traits which they know have strong association with genomic variation. So this is on chromosome 2, and this trait is on chromosome 5, and respectively chromosome 4 and chromosome 7. And you can read actual publications about all these traits. They gave different weight to these traits, and based on this, they estimate this so-called hipster index. So here's the instruction to set up a cluster. Let's try to do that. I usually open this cluster in a new tab. So we need Spark to 11. No, let's see. Okay, we need to create this library. So you copy this name. And go to your workspaces, share it, and create a new library here. Previously we used this jar option, but now we need to use this Maven coordinate. And we need to search for this. So here again you change to Maven Central. And we need to search for this variant Spark. I don't know if it's right one or not. And let's check. 002. I don't know. I hope it's the correct one.
That looks like a snapshot jar. Are they asking you to get it from somewhere?
No, but you see they say you should enter this one.
No, enter the whole thing then. Yeah.
I think it will not work, no?
No. Well,
just for this one?
Yeah. just include the minus snapshot jar.
What happened here? Yeah, try to do it on your own. Maybe you will be faster than me.
Yeah, I think maybe you need the whole thing because you need to say which column it comes from. Oh, there it is. That's the one.
I don't know. I tested it last week, but I forgot what exactly.
Yeah, try to select it. Is anyone else finding a different jar? Is it the same one you guys are finding?
Maybe it's in Spark packages, no?
Yeah.
Ah, okay. Let's try it here. No, I still doesn't find it. Now probably it's in Maven Central. Okay, let's write this one. In create library. Yeah, it has a lot of dependencies, so it took me quite a while to load it.
Yeah, so they asked me to put the whole thing, so I was trying.
Yeah, so I had to do this, right?
Yeah, click advanced, yeah, that's a bit more.
So I did it wrong, right?
Also, you have to put the full text.
I need to do this one, right? Okay, I think I recall how to do that. So I need to copy this, because this is not from the central repository, this is from their kind of custom, right?
Mm.
So the source is still Maven coordinates, but you don't put in anything on the coordinates, you put it in a repository, or what's...
Yeah,
Yeah,
so
here you write this one, and then you need
then you have to do the
you
advanced.
paste URL here.
The repository.
Yeah. So did you get the point? So you need to do this. So in coordinates, you copy that full path, and you pass the URL here. Is everyone on this track?
Yeah, that's what then
Okay, let's try to create it and see if it works. Okay, yeah. So I think Hay was 2.1, so we need to create a new cluster, this time 2.2. So what you can do, well, we can keep it attached automatically. You go to your clusters, and you can stop the previous L cluster. And create a new one. And here we need...
Why do we need a new one? Because if you look at the name on the previous, it says variant 2.11, right?
Yeah.
The Spark version should be 2.2.
Oh, okay.
And Scala 2.11, right? Was it?
Yeah.
Yeah, Spark 2.2 and Scala 2.11. And you can name it random forest. So is everyone fine with these steps?
Okay.
Yeah, you can click on the libraries and make sure that the library is attached. It's not so. Yeah, now it's attached. It still has not started. So when you created the cluster, just make sure that this library we have just created is attached to this cluster. And then you can go back to the notebook and attach the notebook to the cluster. And then we can start loading the data. So here the data is stored on Amazon servers. So you just download it.
So is anyone here into genome-wide association studies for their work? A little bit.
Yeah, that's good, and you should learn it. Actually, it works quite good. I don't know, but according to their blog post, it seems like it's very accurate and very fast in comparison to all the other tools. This is a reduced notebook. If you go to the original one, which is posted in that blog post, they also provide different graphics to compare different packages. So you can run Hail on the same data set. They give these instructions, and then you can compare the results between two. But here they use kind of a priori known variants, and they know that these are strongly associated. So, like, it's a good test because.
I was going to ask how much of the improvement is due to using random forest and how much is due to using Hail. But I guess the speed is all Hail, and the random forest is just a better algorithm to treat the data.
You need to check. I don't remember the exact pictures of the comparison, but maybe in speed it's not faster than Hail, but it's more accurate. But I don't know exactly.
So Hail has its own genome-wide association study.
Yeah, but in Hail you just do like simple linear model without random forest. Like you just take your response variable as phenotype and like variance as a predictor. Yeah, so I have some tasks for you, but we can skip it for now. You can do it at home. When everything is imported, we can load it to variant Spark. So here we import this variant Spark and create a Spark context. So this is VCF file here, as you see. So we load this new VCF data. And I think Russ had it open somewhere. So this is that VCF file. It's a little bit different from what you have seen before, because previously we also had all the annotation scores after every genotype, but here they simplify it, so they keep only genotypes. So you cannot use kind of filtering for this VCF file because it's a little bit reduced VCF file. So here we have only genotypes. So this is a homozygote for the reference allele. And we have 1, 0 here, so it's a heterozygote. So one chromosome has one allele, another has another allele. And actually here, it's also one of the variants you can find, it's insertion. So in the reference, we have 5 nucleotides, but in this particular sample, instead of these 5, we have only G. So as you can see, what happened to these samples, there was a deletion. So 4 letters were dropped from that position in that sample. So sometimes you may also have not only single nucleotide polymorphism, but so-called indovariation. So insertion or deletion. So some letters can be inserted or some can be deleted. But this is some extra. So when everything is finished, so you can see here, we also printed the first 10 features, and these are the sample names. And I think they are familiar to you from the previous work. We also had these human genome samples. Next we load the labels, and the labels is basically the phenotypic data. So this is the hipster index we had before, and different phenotypes are coded with 1s and 0 again. So if you have monobroad, you will have 1. If you don't have monobroad, you will have 0, and they have a few columns. That's what I ask you to check here when I said you do. You can check it later. So you can just use the function head. You use dbfs command with the function head and check the head of that file. So next we actually configure this random forest estimation. So we have our feature. This is the VSTF file. We have labels. This is the phenotypic data. And here you can define how many trees you want to estimate. I believe the more the better, but the more it is, the longer it will run. So they suggest to run 1000 trees. Let's do that. Oh, this is yeah, that's correct. So we have created, so it's only kind of parameters. We have not started the analysis yet. So based on these parameters, we will start the analysis here. So here we have importance analysis scheme.
Can you make it one little bit bigger?
Yeah.
Just,
Better now? So this one will take a few minutes, I think, to run. If you have done, if your cluster was set correctly and the library is attached, this should work.
This is not, this is already the random forest.
Yeah. It's,
so it's assigning some kind of importance value to each feature dimension, which is, I guess,
each variant.
Each position.
Yeah. So as you can see, there are many drops running. Oh.
Okay, so this is based on the Gini impurity scores.
Yeah, so, but this is kind of the math part of this. The Gini impurity score is like uncertainty, right? You want it to be as low as possible.
Yeah, so it's a measure of how homogeneous a certain genotype is inside a partition. So yeah, so for each cell, it's the impurity of that cell minus the impurity after the split into left and right cells.
Yeah, so again, as I say, I'm not very confident explaining this part, but so when it takes a data set, it tries to partition it. So if it was like this one, it has only 2 partitions. And I don't know this Gini score, I think it will be 0, no? If it's only one. So you may have this one. Oh, so you don't have any splits here, so you have zeros here. So the Gini score, it will be this just the n. But if you have something more complicated, I think it first splits it by, like, the first will be split by this, and then you also split additional characters, additional variants. So you will have additional split here. And then you have the Gini score for this one. And of course, if you have the more splits you have, the lower your this delta I, which will be the importance score. So for some variants, if they can be split just into two, that will be the highest importance score. If you need to split into many to get separation between these two phenotypes, then we will decrease the importance score. That's the approach. But if you read it, I think you can click on this gene impurity. There is better explanation for this, and then this is the formula they use to estimate importance. Oh, it's still running. Yeah, I think it takes quite a while. On real data set, it will take a lot of time because this one is very simple. It has very few positions.
Yeah, also we're still having this tiny cluster.
Yeah, that's also true. There is, I think it's called Google Developers YouTube channel. They have quite good simple example with Python code to do all this random forest kind of for example. And you can actually check all the this splitting decision tree process. This is slow. I think
it's okay. Maybe just go down the notebook and explain because we have 5 minutes. So just walk through the rest of the notebook.
So here we use only 4 SNPs, but in genomic data, like in human genome, there is some non-random association between different variants. So if we take 2 variants which are located close to each other, because again, and because also of recombination, so if you have position 1 and position 2, so and let's say you have A and C here, and then you have either 1, either 2. If you have A here, you're also very likely to have C here. So there is some random association between different alleles in the genomic data. This is, in simple terms, linkage disequilibrium. So if we have if there is strong selection, for example, this allele is favored, just because this one is closely related to this, it will be also favored, but it's not actual allele which causes the phenotype, just because it's associated with another allele. So that's why we had 4 sites here which code this phenotypic data, but in the association results we will get more sites, because some of the sites they are in linkage disequilibrium with other sites. So there is some non-random association in the genomic data. Is it finished? Yeah. So here you can see this is the chromosome 2, this is the position, and this is the importance, and it decreases here, right? Or it's not sorted. No, I think it's not sorted. Yeah, we sorted here probably. Yeah, so it's sorted by importance. So if you run this SQL code, we will extract the table, and here it is sorted by importance. So we have the strongest association for this position, chromosome 2. Then we have number of sites in chromosome 5. And again, there might be just 1 site in chromosome 5, but because of this association in the genome data, we have some other sites which also significant here. You can use different tools for visualization. We used display functions before. In this notebook, you can use Python, or you can use R if you want. So this is matplotlib function. It's a common way to make plots in Python. And here you see these are these different sites, and this is their importance. So you can see that one site is highly important and the rest decrease, but all of them are considerably important for phenotypic variation. Yeah, you can do the same in R. and you can visualize the same data just with different picture here. So as you see, it's the same as before, but just different representation. So basically, this analysis shows that it correctly detected our real characters located on chromosome 2, 4, 5, 7, and 15.
When I try to run that one, I get says there is no package called natural sort.
Yeah.
Which cell is it?
That one.
This?
Yeah.
Okay.
Oh, it's because R probably needs this library thing. So it's like a R thing. Yeah. So you have to do spark R, I guess, for research.
Will it work if I
try that one as well.
No?
Yeah.
But it worked.
Never mind.
Maybe I installed it before, because when I ran it on my community edition, it worked. Because I have also tested some other parts here. Yeah, so this is not very user-friendly representation, but if you spend quite some time, you can get this kind of plot. So here we used only subset. We only use this data in some neighboring sites, not all the chromosomes. As you saw, we had 2, 4, 5, 7, and 15. But these are the real sites, as you can see here. This is their genomic position. And if you check the name, the first one was 2282 here. If we go up, yeah, this was the one which was the highest importance. So it was detected correctly. And as you can see here, it has also the highest importance. This one also has high importance, but in our case, I think it might be a little lower. So basically, this is the principle. You have a phenotypic data and you check how the phenotypic data can be associated with genomic variation. There are many problems which are not covered here, like as you say, if you have population structure, for example, you can get some association just due to chance, because the populations are different and random processes different in this population, and some allele can get fixed in this population, and for some reason it also has a particular phenotype, but it may be not involved in the phenotype. So after you, even if you find this association, you need to go to the lab and actually do the check. You need to check the gene to see if it really affects the phenotype and how it affects if you, for example, silence the gene, you stop its function and see if it changes the phenotype. Yeah.
Do you know if this implementation can deal with population?
I don't know, they don't say anything if you can correct population structure. Maybe you need to run on each population separately, that would be the solution, but it depends on your sample size, of course.
Or using Atlantis.
You can correct, of course, like that's a standard way to do it, but I don't know if they have a way to correct it here in this package. You can check, I don't know.
Yeah, we have to really understand exactly what they're doing with the random forest, right? Yeah. So,
yeah, so this is their credit. I just kept it here. But as I say, you can go to the top. There is a link to this original blog post, and you can read more here.
okay, thanks.
Yeah, we are almost on time.
So thank you very much. So that's basically the conclusion for today, 05. And the homework assignment, if you want some kind of credit or certificate, is to write the simulation program for the coalescent. You know, even without mutation, but you can add the mutation. So that you can write in Sage Math, just the Python. Just email it to me. And this stuff that Dmitry talked about is like, you know, you do it out of your own love. And we have a Uppsala Big Data meetup that will happen sort of independent of these courses, right? So you guys are welcome to
to be part of it. Anyone can offer to give a talk, and so maybe Capuchin said he might do something on Terraform, which is how to deploy solutions, not just independent of Databricks, in any cloud, AWS or Google or Azure, without worrying about the details of how each cloud is actually laid out. Some kind of meta magic, which I'll definitely go to it. So usually the meetups happen from 6 p.m. to 9 p.m. Sometimes we stay till 11 p.m. but you can leave. So usually it's 3 hours, and then we typically go and have some burgers and beers. So the meetups can be highly engineering focused, or it can be